Welcome to this incredibly exciting webinar where we are talking about clinical trials and what you need to know. And a big thank you to Camp 4, whose crack team is on screen with me. And we'll be talking about a potential clinical trial with their asset, as well as sharing data and helping us all understand the massive amount of work that goes into clinical trials before it even gets anywhere near a human. So I'm going to jump in with just some broad overview slides for people. And before I do that, I just wanted to make sure people had seen this QR code / link, curesyngap1.org/camp4-gloss. And that is to one slide with some vocabulary words on it. So if people want to go to that glossary and have that open for reference, that might be helpful. If you hear other words in this conversation that you're like, what are they talking about? Please put those in the Q&A or the chat. Yeah, in the Q&A. Please put those in the Q&A and we will make sure we add them to future glossaries. So my name is Mike Graglia. I'm the founder and CEO of SRF, also known as Cure Syngap 1. And as I said, I'm really excited to be kicking off this webinar on ASOs. Just going to go over a couple of introductory slides to level set on this topic. So clinical development 101, what do we need to know? Therapies must be approved by government agencies before they get into humans. And then this is why, before they are prescribed and available to be prescribed. And this is really why we do clinical trials, to show the efficacy. Historically, the safety and efficacy. Historically, determining whether these medicines were safe and beneficial was a challenge. We didn't really have best practice. And so a lot of chemicals got on the market that weren't always safe and had some pretty unfortunate side effects. And this is why, again, we need to do clinical trials. There's a strong hope with doctors, families, researchers, patients, that medicines will work. And then this creates what's known as the placebo effect, where you just take a sugar pill and you think something great's happening because you want it so badly. And the placebo effect is real, and it's something that the medical community takes very seriously. And this is why, over time, randomized placebo-controlled trials have become the gold standard, as they help us really measure the benefit and any potential risks and compare them against placebo. So yeah, I won't get ahead of us here, and then just a reminder, before any trials begin, potential medicines are tested in multiple species of animals, generally monkeys and mice. And then there's additional testing in animals to show potential efficacy and or confirm that it does what we think it does, for instance, make more of a protein. So these are just some level setting ideas we wanted to get out of the way. Generally, when you start talking about clinical trials, you hear phase 1, phase 2, and phase 3. This is before it gets into a human. And for the big diseases, the diseases that affect a lot of people and the medicines that we're all familiar with, phase 1 is just initial safety and dosage, putting drugs into healthy people to make sure that everything's fine when you just give this drug to normal people. Phase 2 is putting it in patients and looking for some impact and making sure it's safe in patients. And then phase 3 is the critical trial. It's generally the biggest one, and it's where we make sure that this drug does what it says it does, right? Insert disease here, cure X, what? Now, in rare diseases, for a variety of reasons, this timeline, this model of phase 1, phase 2, phase 3 is collapsed a little bit. Phase 1, 2 are often put together. So early trials are, we look at safety and efficacy, and we sometimes will start in patients. And then phase 2, 3 can also be combined. And really, it's at the end of that phase 2, 3 that we get the approval we all want, where this drug can be marketed broadly. So you're going to see actually on one of my future slides, a number of other ASOs that have already been through clinical trials have had phase 1, 2 combined. And this is not phase half, which I've heard some people call it. This is collapsing the model that we've used for other drugs that affect broad chunks of humanity down into a more sane timeline for a much smaller population like ours. And then the other important thing here to really bury the lead is OLE. This is another very important term. OLE is open label extension. So when you're in a clinical trial, you're going to get either the drug or a placebo, right? Because we need that double blind randomized. Double blind means the doctor who's giving you the drug or the placebo won't even know what it is, right? All these things have numbers on them. They get randomized at the top. Patients get A or B. And then only at the end of the trial do we unmask the data and understand what was happening. So an open label extension is where that is no longer blind. Now you're looking at the label and you know, hey, I'm getting the drug. And open label extensions allow patients in phase 1/2 to continue treatment regardless of if they were on placebo or drug, right? So some people say, well, I don't know if I want to do phase 1/2 because I might get placebo. Well, the good news is if there's an OLE, even after the trial, you would then have access to the drug, and you would know it was a drug because it would be an open label. And that is pretty compelling because this phase 1/2, phase 2, 3 approval is a process that will take significant amount of time, like at least a year, maybe longer. So if you're eager to get your patient on the meds, that's a strong incentive to take part in the trial. Moving on. So why participate in a clinical trial? It's the fastest way to get access to disease-modifying therapies. And as we will hear from the CAMP4 team, there's technology now that actually can modify these diseases. You've heard me say this on the podcast, which, by the way, I'm still going to do. I've just had a rough month. We are really fortunate to actually be having this conversation. There's a lot of rare diseases who are nowhere near a company with the kind of data you're about to see on this webinar. And given the amount of investment and the work on safety, and if you qualify, we think my advice, and this is going to be my standard talking point to everybody, my advice is if you qualify and you can get into a trial, do the trial. Do it for your kid, do it for the community, and do it for all the kids who aren't even diagnosed yet. Also, what's exciting about ASOs is that ASOs are used up as you think of with normal drugs. There are other therapies that, I'm not going to get into other therapies, but the fact ASOs get consumed by the body when they're introduced, and then you need another dose. And I want to emphasize this is a feature, not a bug. What it means is that we can start low, see what happens, and then if we like what we see, we can go up in dose, see what happens. But if we were to put it in and see some kind of effect we don't like, the good news is the drug will be consumed by the body, and we can go to a lower dose next time, right? So there's a lot of language around one and done and gene therapies. I want to just be clear, this is not that. An ASO is a very sophisticated drug that does get used up by the body and eventually gets, you would need more, and that's a good thing. And I want to emphasize the point I just touched on 2 bullets above. We have a diagnosis, and therefore we can participate in trials. Kids out there who have Syngap that are banging their head against the wall and struggling with all the broken systems we're all too familiar with, if they don't have a Syngap diagnosis, they're not going to know about these trials. They're not going to have access to these trials. And that's just another reason that the families who are here that already know what disease they're fighting should really be excited about opportunities like the one we're about to discuss. And so my position is we need to partner with industry on every single trial, every single time. And what's the alternative? I mean, what if you say, no, I don't want to participate in trials. I think it's okay. Well, please go talk to more families. It's not okay, right? Our kids are getting older. Our kids are getting tougher. I don't want to get political, but the current system is not feeding support services for people with disability. So I really do think the best hope for all our kids is to change this disease in their living bodies as fast as we can to improve their ability to function. And I think the way we do that is through disease-modifying therapies. The way we do disease-modifying therapies is through clinical trials. Lecture complete. 2 more slides. What you're seeing here is 2, 4, 6, 8, 9 different links to 9 different trials, phase 1/2s and phase 3s. These are all trials that are happening or are about to happen in the case of Dravet's phase 3. And the names above those are the companies running them. Stoke and Praxis you've heard about. So they're both also working on Syngap in addition to CAMP4. Ultragenic, Ionis, and Taysha are not working on Syngap, but all of these are trials of antisense oligos that have been dosed in humans and are, I think, to my knowledge, all moving forward, right? They've all generated results that people are excited about. So while this is a first for Syngap, if it happens on schedule, it's not a first for rare disease. There have been at least 5 other ASOs that we know about for 4 other diseases, because Angelman got 2. And hopefully we'll be able to put Syngap on this list really fast. And then before I give it to the CAMP4 team, I just want to talk about hygiene for being involved in trials. Hygiene is just how I think about, and I just added this slide. So if CAMP4, if your lawyers don't like this slide, it's entirely my fault. I'm sorry. But just really quickly, when there's a trial, a lot of people are watching. Companies are watching, patients are watching, patient advocacy groups are watching. And so companies, PAGs, leaders, and patients, we all need to be careful about what we say. And you're about to see that today when CAMP4 talks, right? CAMP4 is a publicly traded company, so they need to worry about regulators. And they're trying to get drugs approved by the FDA and the Europeans and others. So they really need to be thoughtful about a lot of rules and regulations from both the financial side and the medical side. PAGs, our job is to help CAMP4, but also to help Stoke and Praxis and everybody else. And we want all the companies to work on Syngap and get to the finish line. So we also need to think about our primary audience, which is you, the patients. So we really need to consider these things when we talk. If you hear us thinking about what we're saying, that's why. Leaders, I just got a call from a friend of mine who leads another PAG, and he said, there's 2 trials, and I don't know which one I'm going to do, and people are going to ask me, and I don't know what to do and what to say. And I thought, well, I might have that problem one day too. And I think it's fair for a PAG leader to say, this decision about what drug I put in my kid is between me and my family and my spouse, who is not a PAG leader. And so my official answer on what trial I'm doing is no comment, because it's there, we give a lot of our lives to the community, but when it comes to something like this, I think the appropriate professional response is to not comment on which trial we're in, because that is a decision that affects the broader family, and spouses get a vote. And then last but not least, patients, right? We need to remember that the steer for you guys, and this is a year premature, but I just want to start saying it now. If you're in a trial and it's a randomized placebo-controlled trial, you do not want to be sharing on Facebook. Oh my gosh, they just did X. I don't think they've done X before. I think it's working. Or, oh my gosh, they just had a seizure. This new drug is bad. Like, you just no comment is the right answer, because the hygiene is we don't want to influence other people's observations, right? And we don't know if we're on drug or placebo. Now, a few of you might be like, well, wait, all those people on Ravicti were posting like crazy. Oh, Ravicti's working, blah, blah, blah. Well, they knew they were on Ravicti, right? That was not a placebo-controlled trial. That was an open label trial. But the hygiene for a trial like this would be to focus. So with that said, I am going to hand it over to our friends from CAMP4, and I will let you guys introduce yourselves. Thank you for listening, and welcome to the CAMP4 team, and thank you for being here.
So my name is Caleb Moore. I'm the Chief Business Operations Officer at CAMP4. And I really want to say thanks to Mike and the SRF team for hosting this webinar, and more broadly, just for the collaboration and the partnership that we've had as we've navigated the Syngap world. I have to tell you that this program, in a lot of ways, wouldn't exist without SRF. We met Mike at AES in 2022 in Nashville when he saw a poster by one of our scientists and said, I have iPSCs and you need to have them. And that was really a pivotal moment for us in advancing this from a research project to a full-blown program that we're advancing towards the clinic. And so SRF has already been instrumental in the advancement of this program, and we really look forward to partnering going forward. Let's see, my colleagues and I are going to make some forward-looking statements, so please bear with us. Mike gave the appropriate qualifiers of us as a public company. And I'd like to just tell you a little bit about CAMP4 as a company. So we're pioneering a new class of medicines to increase gene expression. And more specifically, we are pioneering the discovery and development of regulatory RNA targeting antisense oligonucleotides, or ASO-based therapies to upregulate gene expression and restore healthy protein levels. That is a mouthful, admittedly. And our whole goal in participating in this webinar is really to help make sense of that and simplify it in a way that's really digestible and understandable to the broader SRF and Syngap community. And really, our special sauce is our understanding of the regulatory RNAs that our medicines target and the role that they play in gene expression. Our goal is really simple. We want to increase Syngap expression or make more Syngap protein. So myself, Yuri, and Dan are speaking on behalf of roughly 45 employees at Camp 4. Our company's headquartered in Kendall Square in Cambridge, Massachusetts. And the founding science comes out of the Whitehead Institute, Boston Children's Hospital, and Harvard Medical School. So what is an ASO? The term gets thrown around a lot, and there's lots of different terms that are used to describe the same thing. So ASO stands for antisense oligonucleotide. People call them ASOs, people call them ASOs, people call them oligos, nucleic acid therapies, all the same thing. These are small nucleic acid molecules that can be engineered to target specific sequences of RNA. And they can fine-tune genetic instructions, helping regulate production of a specific protein. In our case, at Camp 4, we're developing an ASO that increases expression of the SYNGAP1 gene to near normal levels, improves how neurons transmit signals, and alleviates the symptoms of SYNGAP1 patients. We're using very standard ASO chemistry, very much like Spinraza from Biogen, which is approved for spinal muscular atrophy, or SMA. And I mention this point just because the chemical background or the chemical backbone of our medicine is nothing new. It's been tried and true. So we like to say that we're pointing a known tool at a new target. Again, coming back to the idea that regulatory RNAs and the understanding of their role in gene expression is really our special sauce. Mike gave a mention to this up front, but we thought it'd be helpful just to kind of situate ASOs relative to some of the other types of medicines that are typically talked about in the context of CNS disorders. So from left to right, we've highlighted small molecules, gene therapy, and then ASOs. And small molecules are what are the medicines that most of you are likely familiar with. These are pills, small compounds. They're given or they're administered orally. You take them by mouth, right? They go to the gut where they're absorbed or into the bloodstream and can be absorbed throughout the body. And so some gets to the tissues that are intended and then gets processed by the liver and excreted. The role of small molecules is really to bind proteins or receptors and elicit some sort of effect on signaling pathways. I mentioned that they get excreted, so you've got to take them typically daily or repeatedly. Next, if you move to the right-hand column, you've got gene therapy, which Mike mentioned. And again, this is a synthetic gene that is delivered via a virus or a viral vector. And gene therapy is shuttled directly to the brain. We've listed the frequency here as 1 time because one thing that the body or our immune systems is really good at is actually how to handle or deal with a virus. So you can only deliver the therapy 1 time. And importantly, the dose can't be washed out or adjusted. It's very much a permanent solution. And then on the far right side, again, we've talked about ASOs. These are short nucleic acid molecules that target very specific RNA sequences. And these are delivered via intrathecal injection, which we'll talk about. But importantly, we want our ASO medicine to reach the cells that matter in SYNGAP. So it's injected into the spinal cord, it travels directly to the brain, and that's intrathecal delivery or intrathecal injection. The ASOs are delivered or given repeatedly on a monthly or quarterly basis, which we're still working out. And importantly, as Mike mentioned, one of the features of ASO therapies is that you can start low and titrate up. The dose can be adjusted depending on the effects that you're seeing in patients as you progress the therapy. ASOs that target RNA have been around for a while, and the benefits of ASOs have been shown with multiple approved medicines. This slide is intended to communicate that point, and on the left-hand panel, you see what we refer to as the central dogma of biology. Right, DNA makes RNA, makes protein, and ASOs can target pre-mRNA or mRNA in a very specific way to elicit effects on gene expression and protein production. Again, in our case, we're targeting regulatory RNAs and increasing expression of Syngap1 in this case. On the right-hand side, you see a laundry list of antisense oligonucleotides or ASOs that have been approved. You can see the delivery mechanism, how they're actually administered, and then the target tissue that they're intended to reach. You'll note that approved ASOs for other CNS diseases are also delivered by intrathecal injection. And in fact, more than 14,000 patients have been treated with Biogen Spinraza for spinal muscular atrophy. I'm going to pass it over to my colleague Yuri to talk a little bit more about intrathecal delivery and our mechanism of action.
Thanks, Caleb, and thank you to SRF for having us. It's been a real pleasure to get to know SRF, and I've had the great pleasure to meet a number of Syngapians through your organization. So, as Caleb mentioned, one of the important things for any given disease is to make sure that we get the potential medicine to the organ or the places that are most important to correct the underlying root cause. And intrathecal administration of ASOs is required to reach target cells in the brain in the concentration that we need them to get. So an ASO for Syngap patients would theoretically then be administered by intrathecal ejection, also known as a lumbar puncture or spinal tap. IT delivery, as it's called, allows for the ASO then to travel directly to the brain through the fluid, which is running throughout the cerebral spinal sac as well as around the brain. And this has a couple of benefits. One is it results in a more rapid and potent effects compared to other routes of administration. But it also allows the ASO to reach the target cells in the brain while reducing the likelihood of getting large concentrations to other tissues that we do not want to target. So, for example, by delivering through the brain, we are limiting the amount that might get to organs like the liver or the heart, etc. And so all of our brains have a blood-brain barrier, which helps to regulate what gets in and out to the circulation and to the cerebral spinal fluid. And so the blood-brain barrier is something that we need to circumvent to make sure we get the appropriate dose concentration. And this is what the IT approach allows us to do. I just want to underscore what Caleb mentioned, which is after the Spinraza approval, IT administration has become a very common procedure done by neurologists. And so it is also a procedure that's done, for example, in many types of anesthesia. So this is a very common practice. It's a quite safe procedure, and it allows us to actually maximize the benefits while reducing potential risks. And if we go to the next slide, here I'm just going to highlight that because of the effectiveness of using IT, there are a number of antisense oligonucleotides or ASOs that also Mike mentioned earlier that are progressing in clinical trials, specifically for other pediatric developmental epileptic encephalopathies. And so those are listed here. You can see that there's across phase 1, phase 2, as well as phase 3 studies. And I think that this is the type of data that should give us confidence that this is a well-trodden and well studied path that is quite effective and also quite safe, that people are very experienced across institutions around the world. So if we go to the next slide, I think for a haploinsufficient disease like Syngap, there are multiple approaches to treating these disease, and Caleb walked through small molecules, gene therapy, as well as ASOs. And one of the things is that some technologies lend themselves particularly to different types of diseases, a so-called product need fit. And in particular, ASOs and the approach that we're trying to take here at Camp 4, we think really lends itself to haploinsufficient disease because, and Dan will talk more about this, but the regulatory RNAs that we're targeting, we can achieve the levels of gene expression that we think will be meaningful in patients with haploinsufficient disease. And as background in haploinsufficient disease, for most of us, for a given gene, we have two copies, one from each of our parents. And that allows us to get the normal level of protein because each of those copies make or are responsible for being essentially the template for that protein. And so in Syngap, this is definitely the case. And so all of us who do not have Syngap, we have normal Syngap protein levels. However, in a Syngapian or Syngap patient, they will have one healthy allele from one of their parents, and then one usually that makes no or very little protein. And so they essentially have 50% or less. And this is the definition of a haploinsufficient disease. So in the next slide, what we'll see here is how potentially Camp 4's ASOs target. So we have discovered over almost now a decade of work that Caleb summarized that there are these important regulatory elements around genes, so-called reg RNAs, that can allow us to essentially target those controls and increase gene expression from one of those genes. And one way to think about this, for example, is that there are both activators and repressors on given genes that control their function. And we have discovered that there is a particular repressor on and associated with the Syngap transcription and the Syngap gene that we can actually target with our ASO. And we're doing it at the transcriptional level. So that's at the DNA level you can see in the hierarchy that Caleb reviewed with us earlier. There are other approaches such as Fidraza, for example, where they focus more on taking pre-mRNA and then influencing increased splicing or increased processing in between the pre-mRNA to the mRNA. But I think what's important to keep in mind for Camp 4's approach is we're actually targeting at that transcriptional level. And if we go to the next slide, and then we'll just kind of looking forward to some of the data that Dan's going to walk us through, we think this is an approach that really has the potential to essentially take an individual who only has 50% of Syngap protein and bring them to almost near normal levels of Syngap protein, because our Camp 4 ASO would target that repressor, as you can see in the schematic to the right. It would result in displacement of that repressor and then result in increase in gene expression and thus production of protein. And so with that, what I'm excited to do is hand this over to my colleague, Dan, who will now actually walk through some of the preclinical data that we recently presented.
All right, thank you, Yuri. Nice to be here. My name is Dan Tardif. I'm the head of discovery. So my team is really responsible for generating a lot of the early data that really helps us identify the regulatory RNAs and the oligonucleotides that we want to advance towards the clinic. So now we're going to get into a little bit of detail, and I'll hopefully do a good job explaining how it's enabling us to advance towards the clinic. So just as maybe a snapshot of the overall company, we are a clinical stage company. We do have a program in a different rare disease called urea cycle disorders. This is a metabolic disease. And you can see that Syngap is it's in the preclinical stage. And again, have generated some really exciting data to help advance this towards the clinic. Behind this, we have a number of other interests in other neurological diseases, including GBA1 for Parkinson's disease. And then there's a number of other diseases that we're really looking to see where the fit is for our platform to address haploinsufficiencies such as Syngap. So I'll now focus on our Syngap data. So we identified an oligonucleotide sequence that is specifically targeting the regulatory RNA that controls Syngap expression. And our goal is to increase Syngap levels and restore them towards wild type. And in this slide, I'm showing a really nice approach where we can actually look in patient cells to show that there is a decrease in Syngap levels in Syngap patient cells compared to a control, and that we can increase this with oligonucleotide treatment. So these are iPS cell derived neurons. So essentially what we can do is take skin cells from a patient or from a familial control, a sibling or a parent. We can actually take these cells in a dish and turn them into what we call induced pluripotent stem cells. And these are cells that in a dish, you can make them turn into different cell types. And in this case, we can differentiate them into neurons. So neurons are the cell type that express Syngap. So this is very important that we're working in a cell type that's actually dependent on Syngap levels and function. And we can show here, compared to a sibling control, lines that were generated from a patient actually have reduced Syngap level, so they're haploinsufficient. And again, when we add our oligonucleotide that's targeting the regulatory RNA, we see an increase in Syngap levels towards wild type. So this is really nice data, again, showing that we can target the regulatory RNA and that this increases Syngap to a level consistent with wild type healthy Syngap levels. And we can see this across multiple patient lines with different mutations, and it's a very robust effect. And this really sets us up to look in an animal model moving forward. The next slide. So it's very important to go from in vitro, where we're studying cells in a dish, to actually see how does the regulatory RNA control Syngap expression in vivo? How does the oligonucleotide target the regulatory RNA, or does it target the regulatory RNA to increase Syngap in the context of an actual brain in a living animal? So to do this, we turn to a mouse model, and this is what we call a humanized Syngap mouse. And this is a really clever system where Ben Prosser at UPenn, who many people are probably familiar with, had generated this mouse line where the human Syngap gene, including the gene that encodes the protein, but also all the promoter and the regulatory elements that we're targeting with our oligonucleotide, have actually replaced the mouse gene. So there's either two copies of Syngap, human Syngap, and then there's no mouse, or there's a single copy of human Syngap, and again, no mouse. So this really nicely models the haploinsufficient state. And you can see, comparing the first two bars, that when there's a single copy of Syngap, there's about a 50 to 60% reduction. And in this particular study, we administered our ASO directly into the brain. And you can see that there's a nice dose-dependent increase in Syngap protein with increased amount of ASO that we're delivering. So compared to the haploinsufficient untreated, which is in gray, you can see that there's a twofold increase of Syngap protein at the mid and high dose level, and this is essentially indistinguishable from wild type levels. So this is a single dose and working only a couple of weeks later. So this is really nice, showing that the data that we had in vitro, where we had discovered the regulatory RNA, showed that an ASO increased it in human cells. Also, when administered to an animal in the context of a fully wired brain, a full complex tissue, that regulatory RNA controls Syngap expression, and that we can target that regulatory RNA to increase Syngap levels. And the next slide. So that's great. We're increasing Syngap. What does this mean? Okay, for these mice. So there's been a number of studies demonstrating, a number of papers showing that in a regular mouse model of haploinsufficiency, where there's a 50% reduction of regular mouse Syngap, that there's a number of behavioral phenotypes and seizure phenotypes that have been shown to be impacted by Syngap levels. This is a humanized mouse that had not been characterized as much before. So we wanted to, one, ask, is there a defect in various different disease-relevant phenotypes? And then second, does our ASO, when targeting the regulatory RNA and increasing Syngap expression, actually reverse some of these features? So we ran a study where we administered our candidate ASO, and then we looked a few weeks later and assessed these different behaviors. So in these three assays, one is a rotarod. This is looking at sort of motor function, and there's a lot of gait issues and motor function issues in SYNGAP patients, as well as a cognition and memory assay, this Y-maze assay. And then there's this other sort of hyperactivity assay looking at basically how much the mice are moving, and we can really track these. And in all cases, you can see that comparing the 2 copy of SYNGAP in gray and the 1 copy haploinsufficient state in yellow, that there's a significant difference, a deficit in these measures. And after receiving the ASO, there's a reversal of these. All right, so adding the ASO, targeting the regulatory RNA, increasing SYNGAP expression is bringing that back to normal. And on the right, you can see the protein levels. So again, we're administering our oligonucleotide directly into the brain, and you can see that we're increasing SYNGAP very significantly towards wild type levels in both cortex, which is a very sort of general brain region, relevant to a number of SYNGAP phenotypes, as well as hippocampus. So this is very nice sort of linking that we can, yes, we demonstrated we can increase SYNGAP, but this increase in SYNGAP is actually having a beneficial response in a mouse model of haploinsufficiency. And next slide. All right, so this study is very nice. It shows that we're administering when we administer ASO into the brain, we can have this impact. Now, this is different from when we move to the clinic, and we're going to, as Yuri discussed, administering the oligonucleotide through IT injection or intrathecally. So the path of the ASO to the brain is different, whether you're going directly into the brain or whether you're going through the spinal cord. So it's very important for us in a more relevant animal, such as cynomolgus monkey, that we can administer the oligonucleotide intrathecally, that the ASO will go through the CSF, the cerebrospinal fluid, up to the relevant brain regions, engage the regulatory RNA to increase SYNGAP expression. So we ran a study in which we administered 3 different dose levels, a low, mid, and high, and we dosed the monkeys every 2 weeks, and then we sacrificed the animals 2 weeks after, which we have to do to look at the brain and show that we're engaging the target. We also looked at the oligonucleotide in the plasma, and we also looked in the CSF. And the key takeaways from this study are, 1, the oligonucleotide was very well tolerated. We don't have a drug if it's not safe. So we really pay attention to when we administer our oligonucleotide to animals, mice, rats, non-human primates, that it's well tolerated. So that was great to see. We get a sufficient amount of oligonucleotide to all the relevant brain regions, a number of different cortical regions, hippocampus, and I'll show you this in the coming slide. And the amount of oligonucleotide that we got to these brain regions increased SYNGAP expression to a meaningful amount. And I'll walk you through this data now. So here is looking at oligonucleotide concentrations. So the concentration of our drug candidate in multiple different brain regions. So in mouse, I just said cortex, right? It's very small, but in non-human primates, they have a larger brain. So we can look at multiple different brain regions. So the frontal cortex, the side, motor cortex, occipital cortex, which is towards the back. And all of these regions have been linked to some aspect of SYNGAP phenotypes. So we would really want to make sure that we can target all these different regions with oligonucleotide. And what you can see here is the low, mid, and high dose. As we give a higher dose, we get more ASO in the right place, right? So this is what we call a dose dependent or dose linear. So if you give 3 times more ASO, if you administer 3 times more ASO, you get 3 times more ASO in the brain. So this is really important as we look to the future in clinical studies. When we give different dose levels, it's important that the amount of dose correlates with an increase in amount of ASO in tissue. Go to the next slide. Now, of course, what we want to know is that the amount of ASO that we administered and that gets to the brain is having the effect on SYNGAP protein levels. So here what we're showing is a nice dose-dependent increase in SYNGAP protein in all those regions where I showed that we were getting oligonucleotide. So shown on the bottom is sort of where these regions are localized in the brain. And you can see, again, a nice dose-dependent increase across these brain regions. And highlighted at the top is some of the aspects of SYNGAP disease that are impacted, and sort of shown below is what feature of Syngap is relevant. So very excited to show this, that when we administer into the spinal cord space, the ASO gets through the CSF up to the brain, gets to all the relevant brain regions. We get enough of it there so that it increases Syngap levels. Next slide. All right, so what have I told you? We have identified the regulatory RNA that controls Syngap expression. We've identified an oligonucleotide that specifically targets this regulatory RNA and specifically regulates specifically increases Syngap expression. And we can see this in patient iPS-derived neurons where we have Syngap haploinsufficiency. We see this in a humanized Syngap mouse model, again, where this is expressing the human Syngap gene in the context of a whole animal, that it reverses a number of phenotypes, and also that in non-human primates, when we administer through the clinical route of administration by intrathecal injection, we get enough ASO to the right brain regions and increase Syngap levels. And importantly, throughout this, the oligonucleotide has proven to be very well tolerated. And really the next key step for this program is to do formal toxicology studies. There's a very regimented approach to making sure that before any patients see our candidate, that it is considered to be safe and well tolerated. And this is the next step for this program this year. Next slide. And I think Yuri is going to take this.
So thank you, Dan. And as Dan mentioned, so we're initiating our formal what are called GLP toxicology studies. Those are the studies that form the basis of requesting regulatory clearance to start studies in humans. And also, we have been continuing to do work with SRF as well as with clinical experts worldwide around what studies might look like, as well as starting to prepare for studies across multiple regions around the globe. So with that, I will hand it back to Mike or one of the other SRF team members.
I'm here. Thank you very much, Caleb, Dan, and Yuri. And we do have a couple of questions that have piled up in Q&A. And so for the audience, if you don't mind, please type your questions in Q&A and we will get to them. And this is, to be clear, we have Caleb, who is the Chief Business Officer, Dan, who I was just looking up online, completed his PhD 17 years ago, if my math is right. So we have their lead scientists. I didn't have time to look up Yuri, but he probably finished his MD a while ago too. So you've got a doctor, a scientist, business leader, all here. Please do not ask a question, end this webinar, and then email me. Don't do that. This is your time to ask questions. So please go ahead and hit them. And I will just go to the Q&A now. I will start with anonymous attendee. For clinical trial design, I'm assuming that I'm assuming that they mean general anesthesia, would be utilized for these kids getting the IT delivery. Is that correct? Would this be done monthly or quarterly, or has that not been discussed yet?
Yeah, I'll maybe take that. So it's a great important question. I think the good news here is that there, as Caleb and I mentioned, there's lots of experience across pediatric neurologists and epileptologists on giving ASO treatments using intrathecal administration to many different types of patients, and that also includes some of the studies. And so in cases where sedation or anesthesia are needed, then it can be used to make sure that actually everyone is safe, most particularly the patient. In terms of the frequency, so we are continuing to do work here, and at future date, we'll be able to speak more specifically to what the anticipated frequency is. I think the one thing that might be important to keep in mind, and as we talk about a couple of the other questions, Dan can also mention this, is Camp 4's approach is really around unique insight into the biology. we are using chemistry around like the backbone of the chemistry and other features of the ASO that have already been in multiple ASOs that have been approved and given to thousands of patients like Spinraza. So that the good news is it means that we have already a lot of data to pull on as to the characteristics of how the ASO will function and what is the likely dosing regimen as well.
Well, let me echo that back to you, Yuri, just so I can make sure I understood it. I think what Yuri just said is they are not doing anything novel with the ASO. They are using ASO chemistry, where basically the tool they're using has been used in thousands of humans, and we can take comfort from that. That it's the path is well trodden. The novel insight of Camp 4 is that they're pointing that tool, that well-tested tool, at a new place on our gene that helps make more Syngap, right? And that's what we want. So is that right, Yuri? Let me just not.
That's exactly right. That's a great summary. Our approach is we actually don't want to be innovative in terms of how you put the ASO together. We want to take approaches from a tool perspective that have already been tried and true.
So speaking of this unique target that Camp 4 has identified, the next question is, what might be the off-target effects of removing the RNA suppressor?
Yeah, I'm happy to take that one. I think this is actually a very nice aspect of our approach, is that it's very specific. So the regulatory RNAs that are expressed from the regulatory regions of Syngap only work on Syngap. So if we displace a transcription factor from the regulatory RNA in the schematic that Yuri showed, this will only impact the Syngap gene. So if we look at neighboring genes, if we look at other genes relevant to this pathway, they aren't modified. It's really only Syngap that will change its expression level in response to the oligonucleotide. That really enables us to be very precise in how we're targeting this.
Great. Great. And then Beata asked 3 questions, true to form. Are you planning on a placebo trial? If so, she's 4 questions. If so, what ratio of placebo to drug? 2 or 3? What are the ages you might be considering? And 3, are you considering an adult trial? And I want to remind everybody about hygiene. Some of these may not be answerable right now.
I really appreciate this question, and I was scrolling through really great questions that are being posed here, very thoughtful. So while we are not yet able to share a lot of specifics about what our anticipated trial will be, I think there's maybe a couple important things to keep in mind that will be helpful. I think one, as Mike kind of mentioned, we want to make sure that when a study is conducted, that we are running the most rigorous trial that's safe, that gives us all confidence in the result. I think that's really important. That's really important for the regulators who are going to tell us it's okay to start the trial. It's important for clinicians who would eventually decide whether or not they can use this medicine. And I think it's all important for all of you as you think about, would I want to give this to my child or family member? And so because of that, things like placebo can help to make sure that we can have confidence that the data or results that we're seeing are in fact due to the medicine and not due to our hope or anticipation. So I think that's something really important to keep in mind. I think it's also important to keep in mind that if you look at many of the other studies that are being done with ASOs, particularly in other developmental epileptic encephalopathies similar to Syngap, the vast majority of them also are using placebo. So we'll be able to speak to more details, I think, in terms of ages and different portions or segments. I think the other thing to consider is that as part of any overall clinical studies, there are going to be multiple clinical studies. And there might be multiple clinical studies that are focused on different populations of patients who have different characteristics. And so it's also going to be important to be thinking about whether or not one stage of development of the studies might be most appropriate for you and or your child or family member. So I think that's another thing to keep in mind, is that it's not like there's oftentimes a single study, but there might be multiple opportunities, and different families and patients may be better suited based on their circumstance for different stages.
Thank you. All right, from Sarah Driscoll. Dan mentioned that there is a linear correlation in the quantity of ASO administered and what is observed in tissue samples. Is there a similar correlation between the amount of ASO administered and the amount of Syngap rescued? Is there a possibility of overdosing?
Yeah, this is a great question. And if you looked at the data, I think both from the haploinsufficient mouse study as well as the non-human primate study, you kind of notice that with increased dose, you started to plateau off, right? So, we generally believe that there is going to be, maybe a ceiling effect or a maximal amount that will be able to increase SYNGAP. So we would not anticipate, and we have never observed like going fivefold, tenfold, or there's some very significant increase in expression. So that is, really actually one of the key aspects of our platform, is that it enables us to increase within, this physiological range of say twofold, which is really what you want in a haploinsufficiency. So I don't believe that this will be an issue to overdose. I don't think that will happen based on the data that we've generated and our understanding of the mechanism.
And we're currently looking at dosing in, or we're about to start looking at dosing in primates that would give more comfort around what you just
Yeah,
said.
with that repeat dose study, in the non-human primates, again, you saw with 3 doses, the amount of ASO, saw this nice dose linear increase. And between the mid and high dose level, they were pretty similar, right? So there was like a 3-4-fold higher increase in ASO, but the effect on non-human primates was really quite similar.
Now for this next question, I'm going to volunteer to take it for you if you want me to, but if you guys want to take it, would this correct a missense variant?
If you'd like to take it, I'll do okay.
I think I just to remind people about what we say about missense variants, almost all mutations are either protein truncating, nonsense frame shift, stop, or missense. We assume that we believe that all protein truncating mutations act the same way. They render one copy non-functional. But every missense variant, and this is why they're tricky, until we can be sure, is sort of treated as their own special case. So before I even attempt to answer that question, it's hard to answer that question because all missense variants are different. Having said that, if we believe a missense variant acts as a null, in other words, doesn't just makes the copy not work, yes, this should work for them. But if a missense variant makes a protein that actually causes trouble, we don't want to make more of that. And this is why the entire SRF program around missenses exists. This is why we have the model in Finland. This is why we are working with Michael Courtney, because we're trying to figure exactly this out. Given the uncertainty around missenses, I personally, not speaking for Camp 4, would be surprised if they put missenses in the early trials, because they probably want a clean population in the phase 1/2. But hopefully, as we learn more and more about missenses, you could imagine this medicine would come online. We would believe a missense functioned as a null, and maybe we would be able to give it to people. That was Mike talking as SRF, not Mike talk. I do not work for Camp 4. Camp 4, do you guys want to change or amend what I just said?
I might just add one thing. I think just I want to reinforce is that there may be different populations, age, missense, variants, etc., where there might be different types of studies. And some people may have seen, or sometimes, for example, smaller types of studies, and of one type studies, there may be other types of investigator-initiated studies. So I think the thing I would just ask everyone to take away is that sometimes there may be different studies that may be the most appropriate fit, rather than thinking that we need to try and have a one-size-fits-all study.
And with that, I'm going to ask one more question, and then I'm going to let Virginia take over the Q&A because Tony just got home, and I got to make sure that doesn't go badly. But this question is, our daughter was adopted, therefore we do not have healthy familial skin blood, what are the alternatives? I think the assumption here is that to participate, you need to be able to make iPSCs, which was maybe something we weren't clear about. But
Yeah,
now,
I can clarify that. You know, we use the IPSC model as we use that as a model, right? So it told us that yes, there's a difference between there's a significant, 50% reduction in Syngap levels in a familial versus patient situation, and that we can increase Syngap levels towards wild type. We did this over a few different mutations just to see that it was robust and consistent. And this idea of targeting the regulatory RNA could increase SYNGAP in a relevant amount. But that doesn't mean that we're going to use that as sort of part of a clinical trial. Like we need to see an effect in iPS cells before you could receive the drug. This was just a model, sort of experimental system to help give us confidence that we were targeting the regulatory RNA to increase SYNGAP levels. So, yeah.
All right. Next, can you guys hear me? Sorry, I'm in a noisy place.
Yes.
Apologize. Okay, so next one, what is used for patients that get the placebo?
So just very briefly, as I shared, we aren't yet at the ability to kind of share any specifics about what the study design is, but in
In
similar
general.
studies where a placebo is used, typically what they do is they do synthetic CSF fluid. Virginia, would it be okay maybe just because I know we're coming up on time? There's I think been one or more questions just on clinical trial sites and travel.
Yeah, go
Is it
ahead.
okay maybe if I speak to that? I think that's
Yes, please.
also an important practical question many people have.
Yes, please.
So as I was sharing across different study stages, and Mike shared this slide earlier, oftentimes earlier stage studies, so like phase 1 or phase 1/2, tends to have fewer number of participants, and they also, because of that, tend to have a fewer number of sites. As you move into later stage studies, there tends to be more participants and also more sites. I think the important thing is as we are thinking about our plans, we are thinking about the ability where one, this would be in multiple regions around the world. I think two is the other thing is that it is very common to have support in terms of financial support and other logistical support for supporting all the logistics of getting there, because I think we and many others understand how difficult it is on any family to participate, but particularly the challenges of traveling with a Syngapian family member or child. And so I think, again, this is a situation where there may be, depending on one's given circumstance or situation, there might be different stages of trial that might be better given how close or far one might potentially be from some of the given sites as we learn more.
I think we are at time. We still have 10 questions unanswered. Let's see.
how are we doing for time, Camp 4? Do you guys want to plow through them, or do you want us to?
Yeah, I think we can plow through a couple more. I think some of them are, there are some similar ones, so hopefully some of them we've already touched on.
We have several on inclusion exclusion criteria, which I believe we can just answer that right now. We are not able to share that. Is that correct?
Well, we can't give specifics. I think the
Right.
overall guidance would be that different stages of trials tend to have different types of inclusion and exclusion criteria. And so just because maybe one stage of trial may have one set of inclusion and exclusion criteria does not necessarily mean that there wouldn't be opportunity at another one if one didn't qualify.
I think a question I'm seeing a couple times is, so let me just read them. One was said, are there certain variants that will be tested, or are they looking to get a wide range of variants to see how the drug interacts? And then another one said, will the same treatment work for every patient, no matter their variant? Sorry if you have answered this. And this sort of gets back to my protein truncating versus missense, and all protein truncatings are assumed to behave the same. Maybe Dan or Yuri, you guys can say that in a more accessible way, which I think is the answer, right?
Yuri, do you want to take that, or do you want me to?
Yeah, I think the way, I think Dan gave a good description here as well as did you, Mike, but I think we just want to be very thoughtful to make sure that um the either variant or type of mutation lines up with what we're going to be able to measure in that given stage of study. So while we're not again able to speak to specifics present, as we think about and develop over time, we're going to take the best of what we know and the best of what SRF and other experts know to use that to make sure that the stage of study and the criteria would match up best to whatever variant type an individual has.
Thanks. These are getting trickier, I realize. There's an anonymous attendee. Did you also test Syngap protein levels in the temporal lobe?
Yes, and they responded to ASO. So there was an increase in Syngap in the temporal lobe with ASO treatment, comparable to the other regions.
And then this anonymous attendee has some real good questions here that I've asked you guys before. In fact, I think this was the first question I asked you when you told me about the monkeys. Given the monkeys didn't have SYNGAP1 mutation and were wild types, what was their behavior like after receiving the drug?
Right. So when we run these studies, there's a very close observation, both immediately post-dose as well as throughout the duration of studies. And there was no clinical observations made. So there was, whether it was a CSF vehicle control or the ASO, there was no observations made in terms of the monkeys behaving any differently, having any signs or symptoms that they were responding negatively to the ASO. And this was throughout the 6 weeks. And of course, this will be more evaluated in depth during the tox studies that we have planned for later this year. But all is well in terms of the ASO is well tolerated and seems to have no impact
Great.
at the clinical level.
Great, thank you. Marta asked, seems that the distribution in the primate brain is very good. Are you expecting the same in humans?
Yes, we certainly believe that there is a strong correlation in the anatomy of a monkey to a human in terms of the spinal cord and the volume of CSF sort of scale. So we obviously would like to have the same distribution. You administer more ASO to a human than you do a monkey, so that is something that we'll consider moving forward. But we feel that the results that we obtained in the non-human primate study provides optimism for us seeing similar distribution in humans. Of course, you can't measure that, right? So you need to use other surrogates such as oligonucleotide and CSF or other things that people do to look at the exposure of your oligo to the CNS tissue, to brain.
So Suzanne asks, have any other ASOs that are already established and referenced on the slides before used to deliver to missense patients, or know if they're headed in that direction / timeline? Before I let you guys answer that, I want to take the moderator's prerogative here. Just because in Syngap, PTVs, protein truncating variants cause disease, and missenses or we don't know, this varies by disease. This is not like how it works with every single gene. So the question presupposes that we would be translating from a similar model in other diseases, and I want to challenge that assumption. But I think this rolls up under a general phenomenon in our community that the missense patients are sick enough that they get diagnosed, but the mechanism of action and if it is acting as a null or a double negative is unknown. So all these patients are acutely aware that trials like this, they may not be included in early trials. So I don't know if there's anything you want to add to that, or if you want to consider this question covered.
I think so, Mike.
It's two left. One from Sarah Driscoll, which is admittedly a bit forward-looking, but the question is, after FDA approval, how long until until insurance companies get on board with coverage? I will let you answer that if you want. I will only say it is a bit early to maybe talk about that. On the other hand, Virginia is sitting on the screen after having just spent the day on Capitol Hill advocating for exactly this access. So SRF, we are working on advocacy, and we are concerned about making sure that there is coverage for these therapies. But I don't I'd like us to get this in human, and then I think increasingly we will be asking the community to help us with advocacy. Camp 4 or Virginia, do you want to add to that?
Timeline, the timeline is challenging. I don't have a timeline. I don't know. Kim, for if you have a, it's a tricky question to answer right now. Like Mike said, we have to start advocating right now, and that's what we're doing.
It's a very tricky question, and unfortunately, it's an unsatisfying answer of it
Yeah.
depends, and it can change over time.
And then the last question, best for last, how do you plan to measure target engagement in the clinic?
Yes, great question. I think it's really important that, particularly in Syngap, there are multiple what I'm going to call kind of domains of Syngap, and so we're going to need to measure multiple ways as we go into the clinic. So, because this is a approach that really is intended to treat the underlying cause and increase protein, or while there may be some ways to measure target engagement, which may change sooner than others, it's really important that we make sure that we have multiple methods to measure. And this is going to be across all the areas of the condition that you all with children or family members are quite familiar with, in terms of whether it be seizure, whether it be motor, whether it be communication, whether it be social, whether it be behavior. So we're working to be very thoughtful with everyone who's been doing work in this area for a long time, as well as with you all. We want to understand and hear from patients and their family members about how we can best do this as we go into studies in a way that fits into your lives.
Awesome. Thank you. That is the end of the Q&A. We are over time. I am grateful to the team from Camp 4 for sharing your experience and your wisdom with us. Closing thoughts. We are going to do this session again, more time from Camp 4 with our European colleagues. I saw Verena, Jaime, and Katrine on this call, so thanks in advance to Camp 4 for the European version of this and the Australian version of this. Hopefully you'll get just as much engagement. To the families who are here, thank you for coming. This is a pretty strong turnout for a live webinar. I hope this was worth your time. I will tell you that if your doctors, the next time you go see your doctor, say, hey, what do you think about an ASO for Syngap? And if they kind of blink 3 times, give them this YouTube video, because right now there is a non-zero chance, unless you are seen primarily at CHOP or Stanford, that you now know more about ASOs than your doctor. And both of you need to be ramped up. So I think this was a great educational opportunity. I think I've gotten some side text messages while we were here from parents saying, wow, like it suddenly feels very real. And that is the point. I think I hope we are as close to a year away from this being an actual decision we could be making. So it is not premature to think about this and take this seriously. And I just want to thank everyone for the time, to Camp 4 for continued engagement and partnership, and to families. If you're like, wait, what about this? What about this? How do we make this happen? Who's doing this? Guess what? We're a volunteer-driven organization. Call Virginia, volunteer. We have a lot of work to do, and we are excited to keep pushing forward. So if anyone else wants to say anything else, go for it. Otherwise, I'm going to call this one done.
Thank you for having us.
Yeah, thank you. This is great. Thank you for the engagement and the support.
Yeah, thanks a million, Mike and SRF team. We look forward to working with you going forward.
Just one last question. Justin Albrecht slipped it in, which I think is actually a good one to just, would patients need to detox from all seizure meds prior to trials? It's actually
Yeah, this is a great question. I mean, again, we're not providing specific details at this stage yet, but I think if you look at other similar trials, particularly in developmental epileptic encephalopathies, typically in the early stage studies, but even later stage, the goal is typically to start any disease modifying therapy on top of existing anti-epileptics. There may be opportunities later on in the trial where if, for example, seizures and or other domains are improved or better managed, then you can start a process of sometimes withdrawing some of those initial ones. So that is a not uncommon approach in other DEEs. Great question.
Yeah. Thanks again, guys. I'm going to stop before more questions roll in.
All right. Thank you.
Take care.
Thank you.